Hello, I am Stella Biachika. I'm going to demonstrate to you detection of Salmonella in food samples. Specifically, we're going to look at raw milk. We have the milk here. We use a couple of media. The first is an enrichment media, which is buffered peptone water. The second is RVS, or Rappaport Vasiliadis soy broth. And then we have the XLD agar, xylose lysine deoxycholate agar, and brilliant green agar, or what I will call BGA. And then if we have any presumptive Salmonella colonies, we'll confirm them on Simon citrate agar or triple sugar iron agar. So to start with, Salmonella may be present in a food sample, but the cells may be damaged or may be injured, okay? So if we straight away streak it on XLD or BGA, the colonies may not grow. So we start by recovering the cells or resuscitating the cells. So we add the sample to the first enrichment medium, which is the buffered peptone water, or what I'll call BPW. And we add 25 mils. So I use a sterile pipette. I'm going to pick 25 mils of the sample. Make sure that the sample is homogeneous. Always work close to a flame. So I'll pick a sterile pipette. So this is a 5 ml pipette. So I'm going to pipette 5 times from here, and I put in the enrichment broth. Okay, so that is 25. So this is well shaken, and it's incubated at 37 degrees for 24 hours. After the first enrichment in the buffered peptone water at 37 degrees for 24 hours, we're going to shake to make the mixture homogeneous, and then pick 0.1 ml and put in 10 mils of Rappaport Vasiliadis soy broth, or what we call RVS broth. Please note that this RVS is not used in isolation of Salmonella typhi and Salmonella paratyphi, because one of the ingredients, the malachite green, inhibits their growth. So other than RVS, you can use another second enrichment medium that is not RVS, that is called Muller-Kaufmann tetrathionate novobiocin broth, and you can use it alongside the RVS, or you can also use it alone. So after this, after putting the first enrichment, 0.1 ml into the RVS, we are going to incubate this at 41.5 degrees for 24 hours. Following the second enrichment in RVS broth, we are going to homogenize the broth and then streak on a pre-poured plate of XLD and also a pre-poured plate of BGA. We're going to use 2 wire loops concurrently. So you sterilize the wire loops in a flame until they are red hot. Okay, so I'll put one down to cool, and I'll wait for this to cool a bit. I can shake this. So you remember from the streaking video, you divide the bottom of the plate into 4 quadrants. So I'm going to streak in the first quadrant. So I'll just dip this in the media, close, and then I'm going to streak in the first quadrant. Okay. Then I sterilize this again. Cool it. Make sure the wire doesn't touch the surface, and then I'm going to streak from the first quadrant to the second. So I'll make about 3 to 4 streaks, or even 5. Then I'll sterilize this. Always ensure that you sterilize the wire loop in between the streaks. So I'll streak from the second to the third quadrant. Okay. So lastly, in the fourth quadrant, I'll move the streaks from the third. If the wire loop is too hot, you can touch the edges of the media to cool it down. Then streak from the third to the fourth quadrant. Okay, so this is ready for incubation. So I'm also going to streak on BGA. This was on XLD. So following the procedure, the streaking procedure, I'll have to shake this again. Okay, so after the streaking, make sure that you sterilize the wire loops before you keep them to avoid infecting other workers in the lab. So the BGA and XLD are going to be incubated at 37 degrees for 24 hours. After incubation at 37 for 24 hours on XLD and BGA, we examine the plates and look for any presumptive Salmonella colonies. On XLD, they appear as pink colonies with black centers. And then on BGA, they appear as pink white colonies, or they may be red colonies with a red halo around them. So using a sterile wire loop, you pick one of the presumptive colonies. Make sure that the wire loop is cool. You can cool it on the side of the agar. Pick one of the presumptive colonies, and we are going to stab it in what we call, this is a TSI slant, or what we call triple sugar iron agar slant. It's a slant. This part is the slant, and then this lower part is called the butt, B-U-T-T. So we stab the butt with a presumptive colony. Okay, stab the butt, and then remove the wire loop and streak the slant. So we have cells that are going to grow in the butt, which is anaerobic, and then the slant, which is aerobic. Okay, so I sterilize the loop again. Wait for it to cool, or cool it on the side of the agar. And then I'm going to pick another presumptive colony. And this time I am going to stab it in the butt and slant of Simon citrate agar. So what we are doing is a confirmation test to see if the presumptive colonies we have on XLD or BGA are actually Salmonella. So I have stabbed the butt and I'm streaking the slant. Okay. So you always ensure that you sterilize the wire loop after use. So I have done this for colonies that have been picked from XLD. You can confirm as many colonies as possible, but since this is a demonstration, I will only demonstrate with 2. Then the same thing should be done for the colonies picked on BGA. Streak them on Simon citrate and TSI as well. So after the streaking has been done, these are going to be incubated at 37 degrees for 24 hours. After the 24 hours, we shall come and see whether the reactions we have are typical of Salmonella species or not. Okay, after incubation, we now have our slants out. We'll first look at the Simon citrate. The Simon citrate, if the test is negative, there will be no change. So you remember yesterday the slant was green and it's still green now. However, if the Salmonella test is positive, it will change from green to blue. Simon citrate contains citrate as the sole carbon or energy source, and also it contains ammonium as the sole nitrogen source. So what happens when Salmonella is present is it utilizes the ammonium salts, increasing the pH of this medium, turning it from green to blue. Okay, so in this case, this is positive for this is negative for Salmonella, and then this is positive for Salmonella. When we look at the TSI slants, or the triple sugar ion slants, like the name suggests, it contains 3 sugars: lactose, glucose, and sucrose. The glucose is in very small amounts. Salmonella is unable to utilize the lactose and the sucrose. So it will only consume the glucose, which is in very small amount. It's only 0.1%, while the other 2 sugars are 1%. So it will very quickly utilize the glucose, producing acid, making this medium to turn from red to a yellow, as we can see in the bat here. However, because glucose has been utilized, the Salmonella is now going to utilize the nitrogen source. In the presence of oxygen in the slant will turn pinkish, as we can see here. It will turn pinkish, but the bat, because there is very limited oxygen, it will remain yellow. However, because there is production of hydrogen gas, the gas reacts with the sulfates in the medium, producing hydrogen sulfide, which is black. Okay, so that's why we see the black coloration. The reaction can occur very fast, and you at times may miss the yellow bat, and you find only black because of the too much hydrogen gas, or thereafter hydrogen sulfide that has been produced. So in this tube, we have the black, and then the pink here, indicating utilization of the ammonium salts and increasing the pH. So the slant that was originally yellow will turn pink because of increase in pH. So in this case, this is negative for Salmonella, and then these 2 are positive. I've used 2 tubes because they bring out the different scenarios that can occur. There are also other conformation media, not only the Simon citrate and the TSI, but there is also urease agar and other agars that may be present. When you're doing the conformation, it's always advisable to use at least 2 conformation media. If you find that the Simon citrate is indicating positive, while the TSI is indicating negative, you'll have to redo the conformation. Both tests have to agree. Please note that the tests that I've described or demonstrated is purely a detection test. We detect for either presence or absence of Salmonella in a food sample. It's not an enumeration test. And what that means, from our sample, since we have indication of positivity, that means that we would report that Salmonella has been detected in 25 mils of the milk. Remember, we worked with 25 mils of the milk. So that's how we would report the results.